socks on. Sat right in the pee. Dog
All right.
peed on the ground.
We are moving into a new unit, and it's basically this is like the segue into cells. So like, well, yeah, we're gonna talk about how cells divide. We'll get into meiosis. Pretty much nitty gritty stuff. Genetics. I guess the next unit would be genetics if I had to summarize it.
Matt, I'm pretty scared that.
Not exactly sure if I'll finish this lecture today. It is 70 slides. So if not, I guess we'll lecture Monday and go from there. So with cell division, there is a difference between prokaryotic cells and eukaryotic cells. And so with prokaryotic cells, we're looking at bacteria. They produce clones as their byproduct. They don't have a sexual cycle like eukaryotes. And what I mean by sexual cycle, we don't have the mixing of mom and dad DNA to get a slightly different version of an offspring. Growth of bacteria is due to its division and its production of new cells. Because remember, cells want to stay small to maximize that surface area to volume ratio. And if a bacteria cell gets too big, well, then it can't carry out all the functions that it needs to do in a timely manner. So one way to solve that is to pinch into two cells. And so here we have our cell. We'll have to talk a little bit about how bacteria cells replicate, and then it just elongates. The DNA goes to opposite ends. It pinches in two. You get two identical daughter cells or clones. So the technical term for how bacteria replicate is binary fission. And what happens is our DNA in bacteria is circular. So it's not linear like it is in eukaryotes. So we'll talk about linear chromosomes here today. But in bacteria, it's circular. It's packaged into a nucleoid. Remember, bacteria do not have a nucleus. But once that DNA is replicated, it is partitioned, or they migrate to opposite ends of the cell. So that way, each new daughter cell has the same DNA. Now, proteins do control the separation and septum formation. So we'll talk about which proteins are involved in the circular replication. And then we'll also talk about the proteins that are involved in pinching the cell into two. So septum is that starting of the pinch in the cell. So it begins with the replication of DNA. It proceeds bidirectionally. So if this is my circular DNA, it starts at a point of origin, and proteins go this way, and they'll meet, and then you'll have two of the same copies, same circular DNA. Then the cell grows or elongates. So the DNA migrate to opposite ends. We start to elongate, and then the next step will be that septum formation, which is responsible by a protein called FTSZ. So septation in prokaryotic cell division, the formation of the septum, which is basically where the new membrane is being formed to separate the two cells. Usually the midpoint of the cell, responsible by those FTSZ proteins. So a bunch of these proteins, FTSZ, they accumulate midpoint of the cell, and they act like a belt. Basically, the belt tightens, and it literally pinches the cells into two. So here's a diagram showing this FTSZ ring, about midpoint. And you can see that they shortened and pinched the cell into two new cells. This diagram actually shows the formation of the septum. They've dyed it, obviously, for various reasons. Now, eukaryotic chromosomes are a little bit different. They're not circular, they're linear. So the first person to kind of observe these chromosomes, Walter Fleming, German embryologist, looking at cells divided underneath, and he noticed these minute threads, these tiny little threads within the nuclei, and they were divided lengthwise, not circular like in bacteria. And so the word mitosis, which is what we're going to get to hopefully today, means thread. So it's like the splitting of these threads into their new daughter cells. So chromosome numbers do vary among species. Humans, we have the 46 chromosomes, or 23 nearly identical pairs. Carp have 104. I think fruit flies have 6. And so the numbers don't correlate to intelligence or anything like that. Okay, so
males have a bunch?
I don't know.
Probably like 5,000.
No, not 5,000.
No, I know. I think it's like in the hundreds, I thought. I'll look it up.
All right, the possession of these chromosomes is essential to survival, which means that if an embryo is missing a chromosome, we call it a monosomy, which means that it doesn't have the correct number. They usually don't make it to full term and die. If you have an extra copy, depending on where that extra copy is, we call it a trisomy, but it's usually fatal except for the smallest chromosomes involved. And so the smallest chromosomes in the humans are towards the end, like chromosome 20, 21, 22. Structure of a chromosome, kind of complex. DNA is not just DNA. DNA is also wrapped up with proteins. And so chromatin is DNA and protein. It's more protein than DNA when you look at chromatin. A single chromosome contains 140 million nucleotides. And there are certain areas on the chromosome that are active and certain areas of the chromosome that are not active. And so those that are not active, we call them heterochromatin, which means that genes at that segment on the chromosome are not being expressed. Euchromatin means that they are being expressed. So this next diagram shows the difference of how condensed and how open they are. So here with euchromatin, areas being expressed, you can see that it's really, really spread out. Okay, it's not as condensed, so it's less condensed. It's gene-rich. It allows proteins to latch on and to read these genes and make those proteins. So it's active. Heterochromatin means that it's highly condensed. You notice it's all squished together here. Proteins can't come in and bind to it because it's just too crowded. And so these genes are temporarily shut off. And so during different times in an organism's lifetime, certain areas will turn on and off. And so if it's on, it's euchromatin. If it's off, it's heterochromatin. So I think next week we'll do a karyotype lab, and you'll have to match up some chromosomes based on their bands. And so the bands that you're looking at are areas of euchromatin and heterochromatin. Okay, so like I said, chromatin is DNA and proteins. And so DNA kind of looks like a bunch of string of beads, basically. Every 200 nucleotides, it's coiled around some histone proteins. And these histone proteins are made up of arginine and lysine. Now, these two amino acids, which make up that protein, have a positive charge. DNA has a negative charge. And so these two they just stick to each other due to these electrostatic differences. When you get that DNA plus that histone, then you get something called a nucleosome. So a lot of vocab words out here. So DNA is wrapped around histone proteins. Proteins are positive, the DNA is negative, and then when you get DNA and histone together, we call it a nucleosome. So this next slide.
Did you not put the words on there on purpose?
There are active notes on there.
Okay.
You can go back and fill it in.
All right.
I think people are not utilized. I don't know. I'm just not happy with where it's going. So changing it up a bit. So here's the chromosome structure. Double helix. And if we just, zoom in here, here's that double helix, and now it's being wrapped around some proteins. Okay, so now we have chromatin. DNA and protein is a chromatin. And then this is supercoiled together, and it's a part of this segment which is supercoiled together, and it's even further condensed. And so basically a chromosome is just DNA wrapped up super, super tight. And the protein's responsible for all this condensed, I don't want to say condensation, that's not the word I want, but just this condensed area, are the proteins called condensin, which means that they just condense the DNA so it can fit inside a cell. I'm sure you've heard that if you were to take your DNA, just like your strand of DNA, and stretch it out, it would be over 9 feet long. You have that ability to do that. And so how do you get 9 feet of information into a microscopic cell? You have to super condense it to fit in there. So just to recap, okay, here's our chromatin, which is DNA and protein. And if I just take a strand out, you see these solenoids with the nucleosome, but eventually you get to the histone and then the DNA double helix. This is what makes up our chromosomes. So when we talk about mitosis, we're going to talk about the different phases of mitosis. We're actually going to look at cells underneath a microscope and identify stages of mitosis like interphase, prophase, metaphase, anaphase, and telophase. But one of the key parts of interphase is looking for
chromatin in this kind of spaghetti-like state. And then when cells are ready to divide, you'll see them in this condensed chromosome shape. So it goes from spaghetti to X's in eukaryotic cells. So how we study chromosomes is you can actually take a picture of all the chromosomes inside a cell and kind of line them up and look at, and notice that they are different from each other by size. Like I said, in in humans, chromosomes like 1 through 15 are pretty big. Okay, chromosome 1 is the largest. And then like chromosome 22 and 23, they're small. Not 23 because that's the sex pair, but 22, they're really, really small. So chromosomes do vary in size. They do vary in different staining properties. That's the chromatin and the heterochromatin. The location of the centromere. So what I mean by centromere is if this is my really crappy chromosome, the little dot that usually is depicted in cartoons, it can vary. Sometimes it's super up high, sometimes it's smack dab in the middle. The length of each arm, you'll notice differences, and then the positions of the constricted regions along the arms. And so again, you'll see that with your karyotype lab sometime next week. They can also vary in the number of chromosomes inside a cell. And so if they have half the number of chromosomes, we call it haploid. And if they have the correct or normal number of chromosomes, they are diploid. So in humans, in our diploid state, we have 46. But in our haploid state, it's 23. So it's haploid is half the number. If I was looking at carp, it's 104. What's half of 104? 52. So the haploid number in carp would be 52. And so the diploid number reflects the equal genetic contribution from mom and dad to make that offspring. So you get 23 chromosomes from your mom, 23 chromosomes from your dad, and that's how you get 46. So here's a picture of a karyotype, and it can actually tell you quite a bit of information. This is human chromosomes, okay? And you can see that chromosomes 1, 2, 3, they're huge. And then as you go through, they get smaller. So here's chromosome 22. Now, usually on a karyotype picture, they give you some information. 47, XY plus 21. So this, what it's telling you is that, okay, we have 47 chromosomes. Now, is that normal for humans? No, okay? This person is male, because we have a Y chromosome and an X chromosome. So if it was female, it would be XX, and the chromosomes would be the same size.
Is that like Down syndrome?
Yes, it is. And then this plus 21 is saying, hey, you have an extra chromosome on the 21st chromosome. And yes, this is the characteristic of Down syndrome. Women that are about to have children, you can get a karyotype if you're really nervous about, the number of chromosomes inside your baby. There is a procedure where they can get inside, either that way, or they can take a really long syringe and they just scrape off some of the cells from the developing baby. Not scrape, but take the ones that are exfoliated, and then they squash it and magnify and amplify it, and then there's someone that actually sorts them. So that they take a picture of all the chromosomes and then they cut them out, and then they use a computer to sort them, and then they can tell you, yeah, 46, or okay, we have
42.
Yeah, well, they wouldn't have the term, but yeah. Okay, so chromosome karyotypes. You notice that on the picture, I guess I should go back. You notice that they are paired up, and so these are actually homologous chromosomes. Okay. So homologous means a pair of the same kind of chromosomes in a diploid cell, which means that one half is mom and the other half is dad, so maternal and paternal chromosomes. Homolog is a pair of chromosomes of the same kind located in a diploid cell, copy of a pair of homologs come from each gamete and form the zygote. So you can use these terms interchangeably, but just know that they pretty much mean the same thing. You got one side or one chromosome from mom, the other one's from dad. They're paired together. You have a homolog or a homologous pair of chromosomes. So here's a picture just showing that both of these are homologous chromosomes. Like purples to denote one set of parents, we'll say mom, and green is dad. There's that centromere. Now, when your cells are ready to divide, they will replicate each other, okay? And so they'll replicate, and you get a tetrad. So we actually call this a tetrad. When you see two X's or two chromosomes paired up with each other. And each
part of this chromosome is called the sister chromatid. So two sister chromatids make up a chromosome. So again, lots of vocab here.
Wait, so I thought
These are not sister chromatids.
yeah.
But when they're ready to divide, the first step is DNA replication. And so they will replicate themselves, and then they will pair up with mom and dad.
But they just make one like X, right?
This is 1X.
Like when it like in this picture,
Mhm.
it's making like.
Oh yeah, that's this stage. That's this stage.
Oh, okay.
Okay.
So, then they do that a bunch of times.
Right. You lose skin cells. You replace over 5,000 skin cells every single day. Actually, it's probably more than that. So your cells are constantly dividing. This is what they do. They go from this state to this state, and then they go through mitosis. Okay, so chromosome replication. So prior to replication, our chromosomes are arranged in a 30 nanometer fiber, which means that they're super condensed, but then they start to uncondense a little bit. And then after replicating, you get those two identical DNA molecules held together by cohesions. So how they get the nice little X shape is the protein responsible is the cohesion. They become more condensed. They're arranged in a protein scaffolding, which makes them more visible. And then you get your two sister chromatids. The diagram, I think, on the next slide will show the cohesion part. So here is my chromosome, and they replicate, okay? But what holds them together is the cohesion, which are the green little dots. And as it's going through mitosis, which the first phase is prophase and then metaphase, the cohesions start to disappear or condense towards the center until they are ready to be pulled apart. So here's another diagram showing all the cohesions to make sure, yep, we're paired up along each other, we're ready to go. And now we're going through prophase and metaphase, and so the cohesions start to disappear, fall off, until there's only a few left, and then we have our microtubules that kind of pull them apart.
So we don't have worksheets for this one?
I posted it on Google Classroom. You can fill it out. It's not homework. So the overview of eukaryotic cell cycle, 5 phases. You have your G1 phase, your synthesis, and your G2 phase. All 3 of these phases make up interphase. So in the G1 phase, it's called the primary growth phase. It's a huge part of the cell cycle. The cell's just growing, okay? So it hasn't even thought about replication yet. It's just doing what a cell does. And then, if it's ready to start thinking about dividing, well, the first step is that it's going to synthesize or copy its DNA. Because your daughter cell's got to have the same DNA. Then after it's replicated its DNA, we go into the G2 phase, or the gap phase 2, second growth stage. Final preparations. Hey, are we really, really ready to divide? Mitochondria get replicated, because remember, they're not part really of the cell. Like, they have their own set of DNA, so they got to replicate on their own. Chromosomes condense. MTs stand for microtubules. They start to assemble their spindle fibers, and then they start to, try to latch onto these chromosomes.
All parents, office staff, kitchen staff, and janitorial ground staff, please report to the library for a picture at this time.
It's American Educator.
All parents, office staff,
Education
kitchen staff, and janitorial ground staff, meet in the library for a picture. Thank
Take
you.
a picture for the newspaper, and everything's just behind.
So was the chloroplast just like the mitochondria, and that it has to do its own thing too?
I think so, yes. Okay, and then we have our mitosis. So our spindle apparatus assembles. They latch onto those chromosomes. They move the sister chromatids apart. It's a very continuous cycle. There's 5 more stages. You get the prophase, the metaphase, the anaphase, the telophase, and the cytokinesis. So that's the 5 stages. However, in college biology, we're going to talk about another phase, and it's called the pro-metaphase, and it's in between prophase and metaphase. And then finally, you get the cytokinesis, where the cytoplasm divides, and that's why it's called cytokinesis, cyto for cytoplasm cell, creating those 2 new daughter cells. In animal cells, they literally pinch in 2 with the help of actin. In plant cells, because of the cell wall, they have to form a new plate between the dividing cells. So we'll talk more about the differences in cytokinesis with plants and animals. But here's a diagram showing G1, S, G2, and then it goes into mitosis. Now this diagram doesn't have cytokinesis, so really there should be one more tiny little sliver here for cytokinesis. But G1, S, and G2 make up interphase, and that's a big portion of the cell's life is in interphase.
Now you may notice this little G0 phase, right? G0 phase is sometimes a cell just doesn't divide anymore, like it's done dividing. So think of neurons. You can't really regrow back neurons. And there are some cases where it has happened, but neurons are usually hanging out in the G0 phase. So that's what G0 is.
So over here, I like this pie chart a little bit better, I guess. With the G1, yes, the G2, this is all interphase. And then we get into the mitosis, which is the prophase, prometaphase, metaphase, anaphase, telophase. And then I guess they have an extra sliver here. I don't know what that is for. But finally, cytokinesis. And then there's that G0 cell cycle.
So interphase is separate from like the PMAT stuff.
Yes. Okay, now the duration of the cell cycle kind of depends on the cell that you're looking at. In animal embryos, it can happen in under 20 minutes where it goes through the entire cell cycle. Fruit flies, even less than that, about 8 minutes. Mature cells do require time to grow. So some cells, it only takes 24 hours, but our liver cells take over a year to kind of go through that entire cell cycle stage and divide. So like, yes, it is quite possible to regenerate your liver if you have enough of it to begin with. So you may have heard cases of people like, oh, they ruptured their liver, they had to remove part of their liver, but they had enough and they were okay, they needed a transplant. Well, eventually over time, those cells might come back.
So liver is one where you have, is that one where you have two, you only need one? No.
There's,
That's your kidney.
Kidneys, you only need one?
yeah,
But
you have like
liver, you can,
lobes.
like-
You have lobes of liver. I think you have like 3 lobes of liver.
Because you can like donate a chunk of your liver. So could that happen like with your brain?
With your brain.
Like where you can like have
Well, like I said, neuron cells, a lot, they hang out in
That's why
G0
you
phase.
hit your head,
and they don't-
you can't get those
They don't
back.
come back. So at 25, you pretty much have the maximum number of neurons, and then they start to die off. So that's why you need to like keep yourself engaged. Do crossword puzzles. Do things where you actually have to think. You know.
Well, you know when you hit your head and you get like kind of like amnesia only for like a certain period of time. How does that like? How do you like?
I think with that, it has a lot to do with the disruptions and the connections to certain parts of the brain. So they have to reestablish themselves. And it's probably also maybe like inflammation, maybe it's disrupted. Because a lot of times when you hit your brain, you start to produce like toxins, and they may interfere with how neurons communicate with one another, each other. There's a new study out about concussions. Because like when you get a concussion, you get some bruising, but then your brain also produces some toxins. And there's a case study out there that said, you know what the best thing to do for your brain, like after you have a concussion, is to like exercise. No, to exercise. Now maybe not like they did treadmill running. I probably wouldn't recommend that because you're like, you know. But bicycle, anything to kind of help get blood to your brain and push out those toxins.
Play more football.
Okay.
Like, I actually had a serious question. Oh, what's the organ that, like, they say they don't really know what it's for? Pancreas?
The appendix.
Appendix. Oh, yeah, pancreas.
Pancreas is really important.
So the
Yeah.
appendix, they don't know.
Or-
They don't
Oh, they
know.
have an idea of what it was used for. Because it harbors bacteria that can break down cellulose. So, like, a lot of animals can't break down cellulose, but those that can, they harbor bacteria inside their digestive tract. So it breaks down cellulose so they can actually use it. So they believe that at one point, humans ate grass or, you know, some form of it. But then, obviously, we turned into hunters. We developed tools to capture our prey. So we kind of went away from that diet. So.
When I was younger, I used to eat grass.
Okay. You know what?
Okay, let's talk about interphase in depth. So remember interphase.
Kitchen staff and janitorial staff, please report to the library at this
They
time.
must not listen very well.
Kitchen staff and janitorial staff,
Nobody wants
please
their picture
report to the
taken.
library.
Like we were waiting for Jim, so he was 8 minutes late. And then there was like no Mrs. Deal, there was no Mrs. Rinky, there was no Mrs. Burger. Like they didn't show up. We just took a picture without them. And I'm like, this is lame. If I just feel like it should be all for one or just none at all. So,
Yeah.
yeah. Okay. Anyways, So G1, S, G2 are part of interphase. Just to recap what each little phase does, the G1 it's a major portion of growth. So the cell just grows. The S phase, super important for DNA replication, and then the G2 phase, chromosomes coil even more tightly, so they're preparing themselves for mitosis. The point of constriction on a chromosome is the centromere, so that's kind of that little circle that you see on cartoon diagrams that holds the chromosomes together. We do have some proteins, some different proteins involved in how the microtubules connect to these centromeres, and so that protein is called the kinetochore. And remember, the centromere location is a characteristic of chromosomes. Sometimes they're high, sometimes they're low, sometimes they're smack dab in the middle. So here's the kinetochore attached to a chromosome. So here's our centromere, and then there's these two disc-like proteins called the kinetochore that allow microtubules to latch on each side of the chromosome, so that way when it's ready to pull away from each other in the phase of anaphase, it does so. Okay, chromosomes coil more tightly in the process of condensation, so they basically become visible. So if you were to look at a cell underneath a microscope, you wouldn't see chromosomes in the S phase, but then in the G2 phase, that's when you start to see them. And you'll notice that when you do your microscope lab sometime next week. Also in this phase, centrioles, which produce those microtubules to attach to the chromosomes, replicate and they migrate to opposite ends or opposite poles of the cell, and then they extend out their tubulin to attach to the chromosomes. So after interphase, after it goes through G1, S, G2, we hit mitosis, and this is where we're going to separate our chromosomes. So during prophase, mitotic apparatus forms. Basically, chromosomes start off as small, and they appear quite bulky at the ends. We have some ribosomal RNA synthesis that slows down and stops because we're going through cell division, so we don't need to make any proteins. We're ready to divide. So the first step in mitosis is prophase. And key characteristics of prophase, you have to look at the spindles and the centrioles. So here's my nucleus, okay? And here are the centrioles. You can kind of see them radiating microtubules out. And what they're getting ready to do is they're waiting for the nuclear membrane to dissolve, and then they're going to latch onto these chromosomes. And you can kind of see the chromosomes in condensed state here, these darkened areas inside the nucleus.
So those circles are
These fuzzy areas right here. These are the centrioles. And then you kind of see things radiating out. Those are the microtubules. And so this arrangement right here, this is called an aster. Then the nuclear envelope breaks down. And so a lot of times in cartoons, they show it as a dashed line. There's my centrioles with the microtubules radiating out, forming that aster. And eventually, these microtubules are going to come in and latch onto the kinetochores. During pro-metaphase, which is in between prophase and metaphase, the chromosomes attach to the spindle fiber. So that's kind of the key characteristic in pro-metaphase. So it follows after the disassembly of the nuclear envelope. Our condensed chromosomes become attached to the microtubules through those kinetochores, and each chromosome, remember, has two kinetochores on either side. So we get the microtubule attachments, and then bipolar attachment is critical. So you have to make sure that the centrioles are on opposite ends of the poles, and that the chromosomes are lined up so that they split to opposite ends of the poles. And the chromosomes start to move towards the center of the cell. We call this jerky motion congression, and they literally do. They kind of like can't even. I'm just not even going to. Anyways, how these chromosomes move is the assembly and disassembly of microtubules. So if you remember when we discussed the cytoskeleton, how we had a positive end and a negative end. And a positive end, if I'm not mistaken, was towards the cell, and the negative end was away from the cell. Well, these microtubules, they disassemble and they reassemble depending on which direction they want to move. And yes, there are motor proteins involved in the movement of these chromosomes during congression. So here's the microtubule, and they disassemble, and they come back over here and reform. And here are the motor proteins that move the chromosome. So So now we've reached metaphase, and the key characteristic in metaphase is that the chromosomes line up in the middle. That's it. Anaphase, away. That's kind of my little saying. Chromosomes are pulled away, or the chromatids are pulled away, depending on if you're looking at mitosis or meiosis. But in anaphase, it is the shortest phase. And so our centromeres split the chromosomes, and now you get two sister chromatids being pulled away from each other. The cohesion proteins disappear. There are two forms of movements. You don't need to know the two forms, but your book mentions them. In anaphase A, the kinetochores are pulled toward the poles. And then in anaphase B, the poles move apart as the microtubules move apart, specifically in catopsins and cytoplasm. So, I don't know. It's just two different ways that they can move away from each other. So I think on my previous diagram, one of these is responsible. Notice that here we have like a motor protein driven, where this is ATP driven motor protein drives the chromosome movement, and over here the microtubulate dissembles like it's attached, but these guys move and then they're responsible for the movement. So there's two different ways the chromosomes can move. That's what it's trying to say. So here's anaphase. They were chromosomes, now they are sister chromatids, and they highlighted pink to show the kinetochores. So away. So when you look underneath the microscope, you're looking for basically, like I always think of two hands. You hold away two hands. After anaphase, you get to telophase, which is two new cells. The nucleuses reform, or two new nucleuses. So the spindle fibers disassemble. Nuclear envelope surrounds each new set of the sister chromatids, and the chromosomes start to uncoil and become spaghetti-like again. So here's prophase. This is probably what it looks like underneath a microscope. Okay. Metaphase, all these chromosomes lined up in the center. Anaphase. Telophase, two new nucleuses. The chromosomes go back to the spaghetti-like structure of it, and you're back to prophase. So in this diagram, they've stained it, the microtubule red, so you can kind of see that the centrioles are forming, and they're moving to opposite ends of the poles. Here are the two centrioles. They pull things away from each other.
So this is obviously just like constantly happening in cells.
Yeah.
So like anaphase or prophase will happen right after interphase will begin right after telophase.
Yes.
So there's cytoplasm.
What's the difference between like anaphase and telophase?
Okay, so
on that one.
well, here you can actually see that there's still sister chromatids, and there's no nucleus. I know that picture doesn't show the nucleus. In telophase, they're no longer X's. They go back to kind of like spaghetti shaped, and you will see in your microscope two new nucleuses. They almost look like, if you ever crack an egg into a frying pan, you'd get a double yolk. That's what it looks like. It's like one egg but two yolks.
And they're back to that like
Cell.
you said, the spaghetti like the prophase.
Yeah. Okay, so then after mitosis, we go into cytokinesis, and this is where the cytoplasmic contents divide. Happens right after telophase. This is where the organelles, hopefully they were replicated earlier in interphase, but they migrate to make sure that everything's equal, and then it will split into two. So cell division is not complete until that cytoplasm has divided. So in animal cells, what pinches the two cells into two is actin. Acts again like a belt, very similar to those FTSZ proteins in bacteria. Filaments slide past each other, the diameter of the belt decreases, basically the cell pinches, forming that cleavage furrow that deepens all the way towards the center of the cell until it's basically two new cells. Plant cells, we have that cell wall, so that's got to work around that. So cell walls are way too rigid for these actin filaments, so what they have to do instead is basically build a new cell plate between the two cells. So this diagram shows the cleavage furrow, with the actin literally starting to pinch it in two, represented by the cartoon diagram. And here's a plant cell, and you can see that these vesicles are helping to help form the new cell plate between the two new plant cells. And so they build a cell wall to divide and complete
cell division.
Now there are some slight differences in fungi and protists. So like for example, sometimes the nuclei are separated during cytokinesis instead of its own separate little phase. When we talk about fungi in semester 2, we'll talk more about their cytokinesis. Now how is the cell cycle? Oh, you know what? I don't know if I'm gonna go on. I think I'm gonna stop it right here, guys. I think this is a good break. So I guess on Monday I'll talk about the control of the cell cycle with MPFs. We'll talk about what happens when the cell cycle gets out of control, which is cancer. We'll talk about apoptosis, and then we'll start an activity, I think, on Monday.
Those jobs that we're just supposed to fill in like the PowerPoints, you know?